So I slightly changed the title of my talk to an easier one or a more difficult one. So first, I'd like to say a few words about myself and my work. So I'm actually from the Department of Automation, which may sound weird to you. So we are basically a machine learning group. And our work basically focuses on two aspects. One is currently single-cell bioinformatics, especially bioinformatics for current and future HCA work, and the other is in machine learning theory, methods, and medical applications. So in China, probably there are more people know of me because of these two books in machine learning. One is my book, and the other is my translation of the statistical learning book. So in terms of single-cell bioinformatics, what we do, for example, we are developing methods for integrating data from different platforms, like different sequencing protocols and even different omics. And we are very interested in studying the information content of cell and cell types. So what are they? And we are developing methods for detecting differential expression of genes between groups, between genotypes, and along trajectory or across some spatial regions. However, due to the theme of this HCA Asia meeting, to me, it's more like a workshop than a scientific conference. So I will not talk about these details, but rather try to ask some questions that we are trying to answer, but for most of them, probably we don't have an answer yet. So let's start. What is the goal of HCA? I think Jay has gave a very good introduction, so I will go quickly. So our journey to understanding life is start from the outside, and then people realize that the genome is a key part in the story. So the Human Genome Project built a reference for all genetic codes, which enables the association between genotypes and phenotypes. However, this association does not tell much about the underlying story, which happens in each cell. So a cell is the venue where the story coded in the genome happens. And thanks to the development of those advanced single-cell technologies, so scientists started this HCA project. So that's why we are here. And the goal is to build a reference on where and how the genetic code works. And so this is from the white book of the HCA. It will generate a large variety of molecular and imaging data across a wide range of modalities and spatial scales. The key reason we want to study this the whole thing at single cell level is because of cell heterogeneity. So from informatics, we want to ask what are the aspects of this heterogeneity across the whole central dogma. So it turned out that there are many. Just list a few that I personally have been able to get some sense out of that. So at DNA level, we have a lot of genomic and epigenomic factors that can vary among cells. And most of us talk about transcriptome level, but there are many scientists who believe that proteomic level are more important, and which result in the phenomes like metabolism and many other aspects. So currently, most of us talk about on gene expression, but even for gene expression, there are many aspects that have not been touched a lot about, for example, alternative splicing, non-coding RNAs, especially microRNAs. Long non-coding RNAs have some been talked a little bit, but microRNAs has not. So remember the goal of building a large variety of molecular and imaging data across a wide range of modalities and spatial scales. So the modality we are currently looking at is still limited. So I would like to compare the HCA with HGP, which also many people think HCA is will be the next big advancement in science after HGP. So from the informatics view, so we are more or less clear about the goal of HGP. So it build an atlas of all human genes, and the fundamental elements are genes. So for HCA, the fundamental elements are cells. And what is a gene? There are some questions about that, but from the informatic view, a gene is a string of DNA. So although it's hard to define which part of a string may be a DNA, maybe a gene or not, but generally in the format is a string of DNA, and with the alphabet of ACGT, and it can be arranged in a one-dimension order. But what is a cell in terms of information? So we have measured a lot of gene expression data and ataxic data, a lot of data. So they give us a very high dimension of features, and people also talk about metadata. So what is a cell? So it's a high dimension of real numbers in, say, 1,000, 10,000 dimensions. So what should it be in the informatics view? And what's the dimension? So we are three-dimensional creature, and we have a history, so we are at least 4 dimensions, and there may be other underlying dimensions. So this is not clear. So and the size, the human genome is about 3 billion nucleotides, and with the basic unit as BP. But about the HCA, we somehow we estimate that the number, the size should be 37 trillion cells and a lot of dimensions. And what is the basic unit? So with this comparison, I would like to say that for HGP, at the beginning of the HGP, scientists knew what they didn't know at that time. But for HCA, we are at the beginning phase of HCA. We actually don't know what we don't know. So that's a huge difference between those two programs. So with that understanding in the background, so I'd like to go very quickly through what happens in China in this field. Friends from the industry gave me this data early this year. So they told me that China has almost 15% of the global single-cell sequencing market in the last year. And we can say this is American, this is basically Europe, and Japan, and other APAC countries, and this is China. And in terms of output, the publications, we can see China has a huge increase from the 2010s, very little, and from the 1960s, we already here. So a lot of publications. So let's look at what are these works on. So they are on 4 aspects: technology development, biological, scientific studies, and atlas building, and bioinformatics. So actually, the Chinese scientists have developed, have contributed a lot in the technology development. We probably all know that the single-cell RNA-seq started from Dr. Fuzhou Tang in 2009, but at that time he was still at UK as a postdoc, and then he quickly, soon he returned to China. And also, we know that Guoji has done some excellent work in this field when he was in Singapore. That was so before they come back to China. So inside China, so from 2012, so scientists from at BGI developed single-cell exome sequencing technology, and scientists in Peking University and hospitals, Fuchoutang, Sony Xie, and Jie Qiao have done a lot on various type of single-cell sequencing technology, including RNA-seq, methylation, and other sequencing at single-cell level. And yesterday we have heard about the new development at the BGI side that is, so in 2012 they have developed a full-length single-cell RNA-seq, and now yesterday we learned that they are doing the multiple omics single-cell sequencing together. And for multiple omics sequencing, so my colleagues, we here at Tsinghua University and Fuchou at Peking University, they developed technologies for doing methylation and RNA-seq at the same, for same cell, and also for spatial transcriptomics. Nai Hejing at Shanghai also has developed their own technique based on imaging. And of course, we have heard the excellent work from Guoji on microwave-seq yesterday. So a lot of technology development, but most of the labs are only users of technology, and they use it on a large variety of scientific questions. And most of the questions are around the reproduction and development. And then the second topic is cancer and immunology, and there are some other medical topics. So this is some statistics on the number of publications. This is the relative proportion. Actually, it should be like this because the total amount is increasing dramatically. So at the very beginning, more labs are working on the reproduction and development study, and now cancer study is getting more and more attention. And there are some representative scientists like Jie Qiao and Peking U group, and well, I'll go quickly. And so, so currently, there are most of them are on transcriptomics, and it was predicted that epigenomics single-cell study will grow. So atlas building, unfortunately, there are not many labs doing atlas building in China, and I think it's similar as in the rest of the world. People are talking a lot about HCA, but actually what you are doing is single-cell biology. I will tell that this is different, and I will skip that. And also we are working with a hospital in Beijing on building the heart cell atlas. We are doing some experimental validation right now, so hopefully the next time I will have something to share with you. And we develop a lot of, so these are all my colleagues in Tsinghua University, and we develop bioinformatic methods. So now I will move on quickly. The key part I want to talk about, that is the relation with current single-cell biology study with the HCA. I think the relation is close, but the difference is also obvious. That is, for the goals are different. So single-cell biology tries to answer particular biological questions, but the HCA tries to build references for future studies. And with that goal, the scientists are satisfied with the important genes that are okay or sufficient for answering your question. But for HCA, we should be interested in all genes that might be important in the future. Although currently we think they may not contribute to your current study. And also the cells, scientists, so if we look into all the great papers on single-cell biology, they select a lot on the cells and genes. So they only keep the cells that are important for their study. But for HCA, we should try to get a representative of the nature. So it's not only selected for your particular question. And the discovery that single-cell studies only are interested in the current question, and HCA, the goal is for general questions rather than particular questions. And the annotation, so it's okay as long as your annotation can make sense to your current readers in your field, but for HCA, we should have understandable and reusable annotation across domains and across time. And technology side, so some labs are trying to combine multiple technologies, but only 1 or 2 or 3 technologies in each single-cell study. But I predict that in the future, HCA, the data will be from multiple technologies. It's hard to say that one technology will be the dominant one. Okay, I will. So in single-cell biology studies, we kind of know what is useful, but in HCA, we don't know what will be useful, but we try to keep everything, just like in the human genome study, so people not only measure the coding genes, but a lot of non-coding genes, although we don't know what is their use yet, but we need to have them. So it's not equivalent. So I think I will skip this one because I don't have time. This is the most important question. But only one sorry, I think 1 minute is okay for that. So remember in the HGP, so before it's completed, there are some announcements that we have completed, let's say, 99% of the genome. But now when we talk about HCA 1.0, what is the percentage? So there's no theory that tells or predicts how far we are from. So there are some theoretical study needed. And so I will skip this. So HCA is important, but how? We should have more strategic discussions, and these are the findings. And I will stop here with the journal that I'm kind of chafing, and maybe we should do something.
Right. Thank you.